The second part of this lab is we're learning about this second type of cell division that's done by organisms that carry out sexual reproduction. So you already learned that cell division can be defined as the process where one cell splits into two new cells. Organisms like yourself, myself, that reproduce sexually are going to use two separate types of cell division. So mitosis, which we've already covered, and now meiosis. Meiosis is basically a process where a single diploid cell is going to basically go through two separate rounds of cell division. And we're going to call those two separate rounds of cell division meiosis 1 and meiosis 2. Now, by the time we get done with the process of meiosis, what we expect to have produced are 4 haploid daughter cells. And each of those haploid daughter cells should have half as much chromosomes, okay, or half the number of chromosomes as the diploid parent cell that we started with. So for example, you and I have 46 chromosomes, or 23 pairs of chromosomes. So if we have 46 chromosomes in our diploid parent cell, by the time we finish the process of meiosis, our haploid gametes should have half as many chromosomes. So that number should go from 46 chromosomes in our diploid parent cell to 23 chromosomes in our haploid sort of daughter cells, our haploid gametes. So these words diploid and haploid, what do those terms mean? Cells that contain paired sets of chromosomes are referred to as diploid, or 2N. So again, our human diploid cells have 23 pairs of chromosomes, right? So we have these paired sets of chromosomes. We have 23 pairs, or 46 total chromosomes. Haploid, or N cells, contain only one single set of chromosomes. So our human haploid cells, which we're also going to call our gametes, are going to contain 23 chromosomes. In humans, the male gametes are going to be called sperm, okay? And each sperm should contain 23 chromosomes. Each sperm should be formed and develop in the testes, okay? So those are the male reproductive organs. In humans, female gametes are going to be called eggs, or ova. Each egg is going to contain 23 chromosomes and should develop in the female reproductive organs, referred to as the ovaries. So then what happens is, when the haploid sperm that contains 23 chromosomes meets and fuses with the haploid egg that also should contain 23 chromosomes, it's going to produce a diploid zygote. That diploid zygote is going to have those 23 pairs of chromosomes, or a grand total of 46 chromosomes. That zygote that is then formed will then begin to go through mitosis, and it's going to multiply hundreds and millions of times, eventually developing into what we think of as a human. Now that process where the sperm and the egg come together, that process, or that union, is referred to as fertilization. So again, words like diploid and haploid, sperm and egg, zygote, fertilization, those are terms that we want students to be familiar with. All right, it's important that you understand sort of the overall process of how this happens at the organismal level. Now, meiosis is going to be very similar to mitosis in that before the cell is ever going to enter division, the chromosomes are going to be replicated during S phase of interphase, okay? Just like mitosis, interphase is going to precede meiosis. So interphase is going to consist of gap 1, it's going to consist of synthesis, it's going to consist of gap 2, with all of pretty much the same events that happened when we talked about interphase before mitosis. Now there are going to be some additional events that we're going to talk about in regards to meiosis, but some of the same sort of steps, some of the same processes, the nucleus breaking down, chromosomes aligning, are going to be somewhat familiar to what you learned when we talked about mitosis. But there are going to be some very distinct differences. Recall that at the end of mitosis, we ended with 2 genetically identical daughter cells, right? Those 2 cells that were produced at the end of mitosis should have been exactly the same. They should have each had 46 chromosomes, and they should have been identical to the parent cell. That is not going to be the case in meiosis. At the end of meiosis, we expect to have 4 genetically unique haploid daughter cells, right? So in order to accomplish that, we are going to have to talk about some differences that occur. So to understand those differences, the first thing we actually have to do is we have to take a closer look at the chromosomes. So we know that from the earlier video of mitosis that chromosomes are usually found when the cell isn't going through cell division in what we refer to as the unduplicated state. So they kind of look like this. So this is a chromosome and this is a different chromosome. When they go through synthesis, our chromosomes are duplicated, right? And so we can see our duplicated chromosomes here, but they're not separate from one another. They're actually physically held together at the centromere region. And so once our chromosomes are duplicated, we refer to those chromosomes, instead of being in the unduplicated state, we have a chromosome in the duplicated state. Each chromosome in the duplicated state is made up of those 2 sister chromatids. So again, we have 1 chromosome. It goes through it during synthesis. The DNA is replicated. And so now, instead of having our chromosome in the unduplicated state, our chromosome is found in the duplicated state. And as we can see, when the chromosome is in its duplicated state, it is made up of those 2 sister chromatids. Now, one of the things that it's important for students to understand is the pairs of chromosomes that we have. So again, if we have 23 pairs of chromosomes, those chromosomes that we have in our individual cells, those originally came from the sperm and the egg. So there's a paternal 1 chromosome and there's a maternal 1 chromosome. So in other words, the sperm provided a number 1 and the egg provided a number 1 chromosome. So in that zygote that was formed, you got a number 1 chromosome from dad and a number 1 chromosome from mom. Now, these chromosome pairs are referred to as homologous chromosomes, and they have the same types of genes on them. So again, maybe we have genes here at this particular location. The location is actually, the gene location is actually called the locus or the loci. These would be genes for eye color. Okay. Maybe the genes down here would be for hair color. Maybe the genes here would be so that you can make the enzyme salivary amylase. Now, sometimes the genes are exactly the same. So maybe you get all genes for brown eye color. Sometimes the genes are different. Sometimes you have different versions of the gene. So you might have gotten a gene for, brown hair from dad and a different pigment, maybe a lighter pigment, a blonde pigment gene from mom in regards to hair color. You'll learn more about sort of the genes and the variations of genes in these sort of alternate forms of genes called alleles when we do the genetics lab and when we learn about genetics during lecture. But here it's very important that you understand that we have these homologous pairs of chromosomes because we're going to use those terms when we talk about what's happening in the different stages of meiosis. Now, both males and females do not have 23 pairs of homologous chromosomes. Again, homologous chromosomes they're going to be about the same length. They're going to have about the same sort of centromere position, and they're going to have genes at similar locations that code for the same character. Again, whether it's eye color, hair color, or a particular enzyme. These would be a pair of homologous chromosomes. Females, the 23rd pair of chromosomes, called your sex chromosomes, females have 2 X's. Males, for their 23rd pair of chromosomes, have an X and a Y chromosome. They're what we kind of consider a mismatched pair. The reason that I refer to them as mismatched is that the Y chromosome is significantly smaller than the X. Okay, so it does not have the same length, nor does it have the same genes on it. So the Y provides the genes that allow for males to develop testes, to develop sort of their male reproductive organs, and therefore be able to develop sperm. Without those genes, testes and sperm can't be formed. And so again, I think it's important that students understand that yes, when we talk about females, that we really do have 23 pairs of homologous chromosomes. But when it comes to males, really, they only have 22 pairs of homologous chromosomes. Those are sort of what we call the first, 22 pairs are called, autosomes. You don't have to know that term. You'll talk more about that in lecture. But that 23rd pair, the sex chromosomes, those chromosomes in females are matched. You have two X's, but in males, it's an X and a Y. And those chromosomes are not homologous because they do vary in males in regards to the X and Y when we look at length or we look at the gene location. So there are going to be some different events that are going to happen when we talk about meiosis that we never learned about when we talked about mitosis. So the first thing we're going to talk about is during meiosis 1, again, we can talk about the fact that there's going to be interphase. So we're going to have gap 1, we're going to have synthesis, and we're going to have gap 2. Again, gap 1, cells sort of doing normal metabolic function. We can talk about synthesis, we're duplicating the chromosomes. We can talk about gap 2, where we're duplicating more organelles, we're increasing the amount of cytoplasm. And once all of those things happen, now we're entering into prophase 1. Now, during prophase 1, what's going to happen, there are going to be some similar events than in what we covered in mitosis. So we can see that the spindle fibers appear. Remember that the spindle fibers are going to be important. They're going to form that mitotic spindle that are going to attach to the chromosomes and eventually pull them apart. We can see that there'd be little centrosomes that are going to be present, and they're going to migrate to opposite poles. Those are going to act like little organizing centers. The nucleus is going to begin to break down here in prophase 1. But an event that did not happen in mitosis that's important in generating genetic diversity that's going to happen here in meiosis is a process called crossing over. And so what happens is during prophase 1 of meiosis 1, the chromosomes are going to the homologous chromosomes, so for example, paternal 1 and maternal 1 are going to pair together. And when they pair together, what's going to happen is that genetic material is going to be exchanged between these non-sister chromatids of these homologous chromosomes. And what that's going to ensure is that the gametes that are going to be formed are going to be genetically different from one another, because the gametes that form are not going to have chromosomes that are identical because of this genetic material exchange that happens in regards to crossing over. So crossing over happens here in prophase 1. There's also a pro-metaphase 1. Again, we don't really hit on this in the lab book. It's not really hit on in the lecture text that we're currently using either. But kind of the same events happen here. The mitotic spindle is forming. It's moving the chromosomes to the center of the cell, to the equator. So you can see that those homologous chromosomes are being sort of moved with the help of the mitotic spindle to the center. The mitotic spindle, I should have mentioned, also is actually those little spindle fibers attach during prometaphase 1 of meiosis 1 as well. Once those events happen, we then move into metaphase 1. So here in metaphase 1, the homologous chromosomes, again, they're lining up in their little pairs. Sometimes these little pairs are called tetrads. They're called tetrads because the homologous chromosomes have 4 sister chromatids total. So tetra means 4. So we can see that the little tetrads, or the little homologous chromosome pairs, are aligning on the metaphase plate. And as they align on the metaphase plate, they do this sort of randomly. And when we say that they do this kind of randomly, what this random alignment is called is independent assortment. Independent assortment, or the random alignment of sort of the paternal and maternal homologous chromosomes on the metaphase plate, is also going to generate genetic diversity. So if we kind of look here, right? So if this is the metaphase plate, so we're in meiosis, we're in metaphase 1 of meiosis 1, you could see that the chromosomes could line up like this, where the paternal chromosomes are on one side and the maternal chromosomes on the other. It's just as likely that when those little tetrads align, that maybe you get a maternal on this side, a paternal on this side for one pair, but for the second pair down here, notice that the maternal is on the other side, right? And so again, it's kind of arbitrary which side, how the chromosomes align. But what this does is it generates genetic diversity. Again, when the chromosomes are eventually the homologous pairs are going to be separated, you could end up with daughter cells that sort of look like this, one favoring more paternal, one favoring one maternal, or we could have more of a mix in regards to the genetic material. So this process called independent assortment, which you're seeing right here, is really what's happening during metaphase 1 of meiosis 1. Then what we do is we move into anaphase 1 of meiosis 1. Here, the spindle fibers, the little mitotic spindle, are going to begin to pull apart the homologous chromosomes. So paternal chromosome 1 is separated from maternal chromosome 1. Paternal chromosome 2 is separated from maternal chromosome 2. So unlike mitosis, where during metaphase we separated sister chromatids, in anaphase 1 of meiosis 1, we are separating homologous chromosomes. Once that happens, what's going to occur during telophase 1, and you can kind of already begin to see this, is that 2 nuclei are going to form, and cytokinesis is also happening at this time as well, and it's going to begin to form our 2 new haploid daughter cells. Each of these daughter cells is haploid. It has half as much genetic material as the parent cell. So this parent cell, remember, it is a diploid, or a 2N cell. It's got 1, 2, 3, 4 chromosomes. By the time we complete cytokinesis 1 of meiosis 1, our 2 daughter haploid cells are going to have half that amount of genetic material. So you can see that each of our daughter cells is only going to have 2 chromosomes. Again, this is just for example. In your human cells, by the time you finish meiosis 1, you would have 2 haploid daughter cells that would each have 23 chromosomes in each. Now, after you complete cytokinesis 1, of meiosis 1, each of the daughter cells is then going to go through meiosis 2. Now note that in between meiosis 1 and meiosis 2, the DNA is not replicated. Okay, so we don't want to do synthesis in between meiosis 1 and meiosis 2. The idea here is we're trying to reduce the amount of chromosome numbers. Now, prophase 2 is going to be extremely brief. So again, it's going to be very similar to kind of the same events that we talked about in sort of prophase of mitosis. So you can see that the nucleus is breaking down. You can see that the mitotic spindle is forming. You can see our little organization centers, the little centrosomes, are beginning to migrate to opposite poles. Again, in prometaphase 2 of meiosis 2, the nuclear envelope is going to completely disappear, and the spindle fibers are going to attach to each of the little chromosomes that you see here. In metaphase 2 of meiosis 2, the sister chromatids the chromosomes are going to be lined up in single file order on the metaphase plate. There's going to be a little spindle fiber that's attached to each of the sister chromatids that make up each of our chromosomes in the duplicated state. As we move into anaphase 2 of meiosis 2, those two little sister chromatids are going to be separated. They're going to be pulled apart from one another as the mitotic spindle shortens. And then as we move into telophase 2 and cytokinesis 2 of meiosis 2, we are going to have our 4 haploid daughter cells. They're all going to be genetically unique thanks to the processes of crossing over and independent assortment. And we're going to see, if you look very closely, that the chromosomes that exist in each of our haploid cells are in that unduplicated state. So here we would have 2 chromosomes in the unduplicated state. Here we'd have 2 chromosomes in the unduplicated state, as well in these 2 daughter cells. Again, these daughter cells should be genetically unique to one another. They are not identical as the cells that were produced through the process of mitosis. Now, what do you typically do, or what will you be doing in regards to procedures for meiosis? Again, what we want students to do is we want you to go through and we want you to simulate and draw the stages of meiosis. So what happens in prophase 1? Draw that out here, right? Draw the breaking down of the nuclear envelope. Draw the homologous chromosomes pairing up, forming those tetrads. Again, you might obviously have a difficult time drawing that process of crossing over, right? But you could draw something maybe on the side of the little circle that looks like this. Draw crossing over so that, again, if you're a very visual learner, you could be writing here, right? Hey, crossing over is happening. But if you draw that little picture of crossing over right next to the circle, then it helps you to really understand what it means when we say crossing over. Same thing when you're doing, for example, metaphase 1 of meiosis 1. Draw independent assortment. Okay, draw the little ways that the chromosomes could arrange. Again, make sure you're taking the time using the lined portion to write out the key events that are happening in each of the stages. There are also subsets of questions in regards to the process of meiosis throughout the lab, or mitosis in regards to the lab. Make sure that you're answering all of those questions. Those questions are really designed to help test your understanding of the content. Once you've gone through and you've either used the beads to model these processes and then drawn out these little images, or you've just gone through and you've drawn the different stages, you then want to make sure that you observe meiosis using the plant cell what do? We use lilium. Most flowering plants have the ability to produce both male and female gametes. The production of these gametes through meiosis takes place in different parts of flowering plants. So the carpel part is sort of the female part. The stamen part is the male part, where the male gametes would be formed. And we can actually look at where those gametes are being developed using microscopy. We can actually stain those cells and look at, what the cells look like at different phases during meiosis. I've put up some of these pictures here in regards to, what the cells would look like in interphase 1, prophase 1, metaphase 1. We also have prophase 2, metaphase 2, anaphase 2, telophase 2 in the lab book. So do make sure that you're taking time and that you are, again, able to identify each of these phases. And not only can you identify them, but you can talk about the important events that are happening in each phase. Again, are the cells diploid or haploid? Are the chromosomes in the duplicated or unduplicated state? When does crossing over happen? When does independent assortment happen? And then finally, it's really important that students understand both similarities and differences when we talk about mitosis and meiosis. So we do provide a Venn diagram where students can test their knowledge, where you can write the key you can write in unique events that are unique to mitosis on this side, events that are unique to meiosis on this side, and then events that are that are done by both are done in both mitosis and meiosis, sort of in this center overlapping portion. Again, I think it's very important that students take the time to do this. This is a great assessment tool that can really be used to help students understand what's happening in meiosis, what's happening in mitosis, and where there are events that are similar, and obviously where there are events that are different.